Hey, it's Glenn Wokenfeld, Mr. W from learn-biology.com or sciencemusicvideos.com. And this is This Week in AP Biology for Teachers. It's about AP Biology topic 6.5, gene regulation, and 6.6, gene expression and development. Um, Before I get started, I just want to say that who are these videos for? They're for you. And who are you? You're an AP biology teacher. Maybe you're a community college teacher. Maybe you even are a professor at a four-year college. And I'm putting together these videos to demonstrate how much your students can learn by using the interactive tutorials on learn-biology.com or sciencemusicvideos.com. So please take a look, leave me comments, ask me questions, and if you're not an AP biology teacher or a college level biology teacher, forward it on to somebody who is. If you're a student, you'll gain a lot from this video too, but this isn't really about the content, it's about how to teach the content. So what is this content? So imagine if you were an instructor, what it would be like if your students came to class knowing what this diagram was all about and able to tell you that number 6 over here was methylated DNA and therefore it was heterochromatin and it was not transcribed, whereas this is a DNA sequence in which the histone proteins associated with the DNA have been acetylated, and because of that, that DNA is called euchromatin. It's able to be transcribed by RNA polymerase, which is what you see right down here. And that explains enormously amazing things, like, for example, this phenomenon that's called X chromosome inactivation that occurs in female mammals. And I'm not going to go into the biology of it now, but it creates this mosaic pattern, and it explains why tortoiseshell cats have the fur pattern that they have. And if that isn't making you curious to learn more about this, well, I don't know what is. But anyhow, if you're a teacher, you already know this, but there's a tutorial that's going to teach your students all about how to explain this. This is a fairly scary diagram. It's about the formation of a transcription initiation complex, which is what you see over here. And the basic idea of this unit is that in eukaryotes, you need a whole variety of mostly protein molecules that are called transcription factors that create an environment, a situation that allows RNA polymerase to bind at the promoter so that genes can be transcribed. And that allows certain possibilities for gene control that leads to gene specialization and differential gene expression. And finally, that shows up in a tremendously important way during embryonic development, where some cells release morphogens. Those are molecules that determine shape and form. And what those do is they get picked up by receptors in other cells, and they act as transcription factors that activate certain genes. So, that's why island cells are expressing one protein, whereas hair cells are expressing other proteins, and your liver cells are expressing other proteins. It's all about differential gene expression, which is about transcription factors, and of course other things as well. So let me get this out of the way over here. Okay, so the College Board, if you're teaching AP Biology, is going to want you to focus on a couple of things, and these are all in these are parts of Unit 6, and Topic 6.5 focuses on differences in gene expression as being key to differential phenotype between organisms and between cells. So it's not just the sequence of A, T, C's, and G's, but it's about how those genes actually wind up getting regulated and expressed. That leads to a whole incredibly amazing topic called epigenetics. So it's the level above the sequence. And that's the thing that I demonstrated before with the diagram. It's about how the DNA actually gets packaged, how tightly it's wound up, that allows it to be expressed or not expressed. So the basic idea, all cells have the same DNA in a eukaryotic organism, but different cells have different epigenetic modifications, and that's leading to differential expression of genes in those other cells. I guess I took care of that next topic when I talked about epigenetic changes and cell differentiation, and that of course plays a key role in development through processes like embryonic induction. Another phenomenon that you'll learn about in this unit is how there can be coordination of gene expression. So, for example, a hormone like estrogen is going to wind up causing changes all throughout the body because there are similar cytoplasmic receptors, and those act as transcription factors, turning genes on, though the genes that they turn on might be different in different tissues. And there are different mechanisms that happen in plants. So that's topic 6.5. And then topic 6.6, again, is more of a focus on transcription factors, how those actually work, how various regulatory molecules work to inhibit or promote instruction, I guess that's what's on my mind because I'm a teacher, how different regulatory molecules can inhibit or promote transcription, and then how gene regulation results in differential gene expression, as we were talking about before. So, and then finally, about small RNAs that are playing a role in controlling transcription and translation, in fact. So, the way that we're going to approach that through the tutorials on learn-biology.com and sciencemusicvideos.com is by two modules. Modules are like units with multiple tutorials inside them. One is about eukaryotic gene regulation, and the other is on animal development. And you know, animal development isn't squarely within the new kind of framework that they have in the 2019 AP Biology course, but nevertheless, it's a great way to tell the story. And of course, if you tell good stories, your students are going to remember the material. So let's go to sciencemusicvideos.com and let's take a look. So I'm actually going to jump back a little bit. So here's our sciencemusicvideos.com main menu. It's the 8 topics that were set up by the College Board in the 2019 redesign. And we're in Unit 6 over here, and we're talking about eukaryotic gene regulation. So I'm clicking that. And that takes us to the unit. And so the first of the 3 tutorials talks about regulation of chromatin structure. And that talks about the difference between heterochromatin, which is this really dark stuff, and euchromatin, which is this light stuff. The heterochromatin is tightly bound up. The euchromatin is relaxed, and therefore the genes are capable of being transcribed. And again, the reason why I'm showing you this is because what will happen when you send your students to sciencemusicvideos.com is they'll do this short reading, and then bam, they're going to be interacting with a quiz. And that quiz will get them to do things like, label things. So let's see. Inactive, untranscribed DNA, that's got to be the dark stuff. So I'll bet you that's got to be this one over here. Fantastic, I got it right. Ribosome assembly site, that's got to be the nucleolus. That's got to be this over here. But notice that we're getting feedback, and I'm going to purposely do one wrong over here. Let's see. Yeah, good. And so you see that you're getting some feedback over here, and that is what my website is all about. It's about giving students feedback as they learn. And that way they're going to wind up developing correct ideas. And again, it's orders of magnitude more effective than a textbook, and we'll talk more about this later, but it's at a tiny fraction of the price. So a great way to tell this story is to talk about X chromosome inactivation and how that leads to this kind of tortoiseshell pattern through the fact that in some lineages of cells, a paternal X chromosome will be active, and others, the maternal X chromosome will be active. Obviously, this is only in females. Males don't have this, but it leads to a kind of mosaic pattern, and that's how you get this tortoiseshell cat thing over here. And again, there's a lot of interaction. There's quizzes that tell you about this. Here's a reading about the other side of epigenetics. This is all epigenetics because this is all about modification of DNA. But another thing that's been recently discovered about epigenetics is about how these modifications can be transferred intergenerationally. So this is a story about the Dutch hunger winter. Amazing stuff. Your students will be amazed as mine were. That leads us to a tutorial about how transcription is regulated. And again, it's going through the basics of transcription and then talking about how that happens in eukaryotes, the whole idea about the excision of introns so that you just have exons. And then it talks about the formation of the transcription initiation complex, super complex stuff. Your students need to interact with it. They'll get to do that interaction on learn-biology.com and sciencemusicvideos.com. This was the example that I was talking to you about before about steroid hormones binding with cytoplasmic receptors. And again, what's the key idea? Holy cow, your students are going to learn so much by this kind of interaction. And again, these are the interesting stories that you can be telling in the lectures that you do in your classroom so that your students will really understand the material. So let's look at the last tutorial in this module. And this talks about post-transcriptional eukaryotic gene regulation. So here's transcription, but then after transcription, what you can have is, for example, let's see if I can make that a little bit bigger. Fantastic. So what you can have is alternative splicing together of exons. So from one pre-mRNA that has 6 exon regions, you can splice that together in various ways where each exon codes for a functional domain, and therefore you have functional proteins, but they're all a little bit different, more diversity. So that's basically the story over there. We're also going to talk about regulation by molecules like like microRNAs that are controlling transcription. And there's even more, though this frankly gets beyond the scope of most AP biology classes, but it's incredible stuff to look at because you can look at what happens with regulation of translation by proteins like ferritin mRNA. So the ferritin mRNA isn't the protein, it's how this molecule winds up regulating ferritin mRNA. So that takes us to the end of this tutorial, and let's move on to the next unit, which is animal development and cell specialization. So again, the curriculum's changed in the 2019 redesign. And of course, if you're just a new AP biology teacher, you've never experienced anything else. But if you've been teaching the curriculum for years, this is an adaptation because it's about cutting content because less is more, and I philosophically agree with this. But nevertheless, you can still tell a great story about gene regulation and expression through telling the story of animal development. My first tutorial goes through some of the basics just so that students understand some of the things that need to be explained. Like after fertilization, cells go through a series of rapid cleavage divisions, but then how do they form into this hollow ball of cells? And it's not important that students know what it is, but it is important to know that they moved. How do they know where to go? And then, of course, there's the formation of the gastrula, gastrulation, the most important event that ever happened in your life. So how does that happen? How do these mesodermal cells, and you don't need to worry about mesoderm, but you just need to know that there are these migrations. How do the migrations happen so that you wind up forming the basic structure of the embryo, which of course for us is a tube within a tube? This is just laying out the basic pattern. It doesn't go into a lot of detail, but it sort of sets what the map is. Then what I do is I run students through a bunch of principles of animal development. And those first principles I'll take you through is that there's genomic equivalence. All of the cells in your body are genetically the same, except for gametes, of course. But they're different because they're expressing different genes. And how do we know about genomic equivalence? Because of cloning. So talk about cloning up at the top. Then more about genomic, how cells are different because they're expressing different genes. Another principle is about the process of determination and differentiation. And again, this is all part of the same theme. Cells are different because they're expressing different genes. How does that happen? Because of signals that they're sending that are telling cells to turn certain genes on and other genes off. And that, of course, is controlling the cell cycle. And then we get into something that is squarely within the standards of the AP biology curriculum, this idea of, well, actually, this one precedes it, because this is like, how do cells know what to do right from the start? Well, they know that because there are different there are different molecules within the egg that are at different concentrations in different parts of the egg, and those molecules themselves act as transcription factors that start activating different genes. But then we get to the next level, which is about induction causing differentiation, and we talk about some of these fantastic experiments that were done in the 1930s. Then we finally end development by talking about more principles, how there are gradients of morphogens that, again, tell cells what they should do based on their position in the organism. So we look at some studies of nematodes and how different tissues are signaled so that they develop in different ways. There's the whole French flag model, how in the development of your hand did your pinky know to become your pinky and your thumb know to become your thumb? Well, that's all about, again, cells knowing what to develop into based on the signals that they're receiving. So basically, like, who do I grow into is kind of based on where I am and what signals I'm receiving. So that's the basic idea there. And then this talks about apoptosis, this incredible process by which tissue gets sculpted away through programmed cell death. How does that happen? We talk about some of the signaling pathways that are involved in that. And then finally, all the way to talking about homeotic genes and how sequential patterns lead to, first the highest level genes are doing things like controlling, where's the anterior portion, where's the posterior portion? And then you have the development of some major body regions and then finer segments, over here, and then the finest segment over here. And then finally, homeotic genes are going to be determining which appendages wind up growing in which segment. I know that I sort of breeze through that at an incredible speed, but what will happen is that your students will get to slowly read this material. And as they read, they'll take it in, they'll take the quizzes, and then just to show you, I wonder if I have it up here, they'll use the student learning guide. And so here's the student learning guide. And as students read, they'll be taking notes, they'll be defining terms, they'll be explaining the diagrams, they'll be summarizing. You won't believe the amount of learning that your students are going to wind up doing on account of the completion of the student learning guide and the completion of the tutorial on sciencemusicvideos.com. So that has taken you through the website, and let me take you back here and show you a couple of other things. There's an incredible resource on HHMI that, and I just can't get enough of HHMI. I think they're the greatest. So they've done a lot with stickleback evolution and the gene regulation that leads to, for example, whether sticklebacks have spines, pelvic spines, or not. And so go to this website, biointeractive.org, classroom resources, search for stickleback, and you'll see a couple of things. First of all, this is taking you to the YouTube link. So this is this fantastic video that is part of a series that's narrated by one of my heroes, Sean Carroll, and this is taking you through the evolution of the stickleback fish. Fantastic stuff. I know that this is really not a particularly good picture of it. Maybe I can find something better. Yeah, there's the stickleback, and it's showing you how these spines develop in the marine forms, but they don't develop in the freshwater forms. That's kind of a form of convergent evolution that happens wherever these animals are confined to the freshwater environments. And they've identified the genes that wind up controlling that, and they're regulatory genes. That's really the whole point of it. And so your students can learn about that through this video. And again, you can do so much. They have this entire stickleback lab at HHMI you can do where you simulate determining whether the fish have spines or not, and then you do a genetic analysis. I think there's even a little bit of chi-square that happens in there, though my students aren't ready for that yet. They will be soon. At the very least, show them the video and then have them do this video quiz. And then there's another great activity that involves gene modeling of, where you look at all the different genes that, you look at which regulatory genes are on, which regulatory genes are off, and how that controls whether the gene is going to be expressed, for example, in the mouth or in the pelvic region. So anyhow, can't recommend HHMI highly enough, and I totally recommend that you do that. So remember who this is for. Use learn-biology.com, and your students are just going to crush it in your course. They're going to learn so much. It's just an incredible resource, and you know, it's for your students, but it's also for you, because it's going to make you so happy because your students are going to learn so much. So again, just want to say thanks. You can start your experience with learn-biology.com or sciencemusicvideos.com with a free trial. You can test drive it, see if you like it, and believe me, you will. You get your students on the site, you will not believe how much they learn. You'll be amazed, you'll be delighted. So please leave me comments, ask me questions, forward this video. I wrote forward them, but really what I meant is forward this video on to other AP college level biology teachers. Thank you so much. Really appreciate your time, really appreciate your support. Want to hear from you. Leave me comments, like this video. You know what to do. Thank you so much.